step to that step and spend all the time in very narrow domain and just working out your synthesis and cranking up some variation of that. Okay. So any question, anything? That's why I was spending a little more time on the practical issues of these organic probes. And you can again ask question today or tomorrow. I would like to spend some time, and unless I get 10 questions, I'm not going to teach you anymore that day. So I like 10 questions from the audience, just 1 minute finish, before I start the next lecture tomorrow. Okay, the students particularly. Professors are welcome to ask the question, yeah.
Is there anything that you can utilize it in chemical engineering?
Yes. Chemical engineering, see, one of the practical things is difference between we were talking about, what is difference between chemistry and chemical engineering? Maybe in this country there isn't, but in the US there is a difference. And the difference is that chemists, they are doing fundamental research, designing the structure and synthesizing and so on.
They are producing in a lab, they are experimenting in a lab and producing a very small, chemical engineering producing a bulb.
That's the difference. The difference is that engineers are looking at applications, motivate development, and one development is a scale up. So she's making some molecule, you're a chemist? Yeah, so she's making something and makes a very tiny amount. And each step has certain yield, right? And you will look through that and say, well, I don't think this is going to be about practical. Too many steps. I'm not saying your research, I'm just giving an example. So you as a chemical engineer, look at the steps, look at cost analysis, look at how you can scale up this material reproducible. Or she got lucky and made it and wrote the paper, but you cannot reproduce it or other people cannot reproduce it exactly. So that's what chemical engineer does, to make those material practical, as you said, by looking at a scale up. Is it easy way to scale up? Is it easy way to process and make a uniform, the same material each time? Is it cheap or enough? Because look, now, can you hear me still? Not well. Okay, so the idea is technology to patient material very pocket, but not with money. I would like to have it with money, but that's okay. So the idea again is cost. This is what is one of the big concern about nano. You make these nano formulations, how extensive a method you need. Is it a disruptive technology, which means you need to start and make this? Is it affordable? If not, then maybe some benefits of nano, but who would want to use it? Patients, and particularly countries like India, you cannot afford a very extensive drug or treatment. So that's what there. So again, open up the dialogue with your chemist friend. Hey, let's work together. I contest how it can be scaled up. Whether it will ever be practical. Yeah.
In one of your slides, you have shown propagation of laser pulse through turbid medium. So, what is the significance of using turbid medium? that
No, no, no. I'm not saying that you use a turbid medium. I said the tissues are turbid media by inherently. They are not uniform. You look at it like this, you're looking at any tissue structures. There are different domains with different kind of cellular structure. So they are inherently turbid. I didn't mean, if I implied, so I gave you an example that if you take an example of a turbid medium and the light propagate, this is the property they will have. I wasn't saying use a turbid medium, so, okay, but thanks for asking clarification. Yes, sir.
Yes, sir. Actually, my question is that if anybody want to know about the dipole moment in a protein ligand interaction, so is it possible to kind of by the help of these techniques?
Oh, of course, of course. So you can probe it in a number of ways. You can probe it because again, using light technique, one of them is imaging, the other one is sensing, right? So sensing, and the third one is you can use a spectroscopy. So let me give you an example of that. So for example, ligand and the protein interaction, you can always do through FRET, right? Fluorescence resonance energy transfer. So protein or ligand absorbs, transfers over here, and you can look at emission is coming. That will give you information about interaction. Okay, the second one is you can do a spectroscopy. So you can do Raman spectroscopy, for example, and if there is interaction, any kind of chemical association, so on, you can look at changes in vibrational spectrum. Okay. Also, certain ligands, when they bind with protein, they change their conformation, so on. Fluorescence intensity may change, or the lifetime may change. So there are all these probes. It depends on individual case what you're looking at, but very rarely you can do that. That's one of the hot areas to look at these interactions. You can also do photolithography, but that's a little more sophisticated. These are much easier to implement. Yes.
The range is in IR, that is near IR, 900 nanometer. So the frequency become very less at that.
Yes.
So how we get the better and better image that you are showing in near IR, we can get better images.
It's not related to energy or frequency. It's related to the photons that get in, the photons come out. You are looking at the photons coming out. You are not looking at their energy, you know. So I don't know why you are confused with that. As I said, that it needs a higher intensity. So it's intensity driven, not energy driven. Yeah, the higher order effects, they depend on the electric field. The electric field is not dependent of light, is not dependent on energy. It dependent on intensity, how intense it is. Okay, any other questions? So think about 10 questions tomorrow. Okay? All right. Let's look at, so if I want to take advantage of, I just want to, again, not give you too much details because most of you are chemists over here or a chemical engineer or biotechnology. Only one I noticed yesterday, physicist. Who is a physicist? Physics. Okay, she's not here today. All right. So anyway, let's move on. So I wanted to emphasize that how you can measure these 6 parameters, location, time, concentration at that location, and the temperature. Okay. And so this is one of the labs, our labs, which is fairly complicated, and I do not envision that you would have this lab in a short time at Allahabad. Okay. But one of the things could be is that you can collaborate if you're making some probe. Sometimes it's easier to collaborate with people who are not at your own institution. They are someplace else. There can be more effective collaboration. I don't know why. But so you could always have an interaction with somebody, another place who may have this kind of setup and would love to have your molecule or probe to use it. So here is a very simple thing, is that in our case, we need to take two beams. We need to take two beams, and two beams can be from the same wavelength, then you can do 2-photon emission. Or they can be shifted so that their difference can be to vibrational band, and that you can use for like vibrational nonlinear spectroscopy called CARS. Okay, so we have a laser source which is picosecond. Then we split it in two parts. On one goes through time delay, so it could be adjusted. Other one goes through a system called optical parametric oscillator that shifts its frequency to another or wavelength to another. And then you combine these two and synchronize with this delay, and then can come together to the sample this way. And this is to scan the beam. Okay? And here the stage can be moved up and down to look at 3D section. So you can image by multimodal thing. Then at a certain point, I can also focus a laser beam, okay, and look at scattered spectrum to get a Raman spectrum of that point. So we can simultaneously imaging using nonlinear techniques and a spectroscopy using this Raman. And the spectroscopy allows us these spectrum. So you take a vibrational band that may be characteristic of protein and is related to the concentration of protein at that point. We calibrate with a standard sample of protein, and then we can get concentration of that. Same point, if we calibrate with lipids spectrum, then we can get concentration of lipid, and we can get DNA and RNA. So we can locally know the concentrations of each component as well as the imaging of the structure. So this is imaging. We use CARS, Coherent Anti-Stokes, vibrational resolve imaging, and Raman spectroscopy each side. Then I can also do 2-photon fluorescence imaging to look at DNA and RNA distribution, because organic dye called acridine orange, when it interacts with DNA, its emission is green. But because DNA is double-stranded. When it goes to RNA, which is single-stranded, its emission is in the red. So by looking at the colors and their distribution, I can see where is DNA and where is RNA. So we can map out too also. So basically, this is the structure that you will get. You cannot see in this contrast image, but you can see this RNA distribution, DNA distribution, protein distribution. You can map out by color coding DNA, RNA, proteins, and lipids by color coding. So you can get this image of the cell, and then I have a dynamic process, something that's changing. We can see how the concentration of each component changes and the distribution changes. For example, we took the cell and we induced apoptosis by giving a chemical something, and the cell gets damaged and then it wants to kill itself, right? So that's the process of apoptosis. So in apoptosis, the cell fragments, starts breaking down. And you can see that 30 minutes after treatment, the cell is still whole, but then after 24 minutes or hours of treatment, they fragment. Their distribution of DNA, protein, and lipids, they'll start changing. Okay, so it gives you a fundamental idea about the process of apoptosis. And you can use this thing also to probe the interaction of drug, how the drug effective the drug is in killing the cell. Okay. Another thing we did was use FRAP, the fluorescence recovery after flow bleaching. And remember, GFP is green fluorescent protein. So we used that protein and bleached it and recovery, and we found that in apoptotic cell, the mobility is considerably reduced. It does not recover very fast. It is a very long life, seconds, still not recovering that region. Whereas non-treated immediately recovers the bleaching because there is a lot of mobility there. Okay, so these are the things that we can study by imaging. Similarly, we're looking at protein distribution after chemotherapy, how the cells, doxorubicin is a very potent anti-cancer drug, is used for chemotherapy. So we showed over here that how we can look at the distribution after drug administration. This is untreated cell. You can look at lipid distribution, protein distribution. When you treat it, you can see the lipids, how it is over here. Changing concentration, and protein, look at how it stops protein synthesis, there. So another one we are doing, what we call the area, we are very excited recently about this idea of liquid biopsy. What is liquid biopsy? It's a diagnosis where you utilize body liquid samples. What are the body liquid samples? Blood, urine, okay, saliva, okay, all these are body fluid, liquid. And they can have signature of diseases too. So this one very active area is how we can take this body fluid and look at signature of a disease, whether it's a cancer or whether it is infection or whatever. Routinely, that's how we do it. But blood analysis, you take blood sample and you try to do some blood analysis of proteins, blood analysis, consider ions, and all these, and sugar, and all these analysis. So spectroscopy can be utilized also to profile biological structure and do liquid biopsy. So what we did was we have developed our hardware and a software that analyzes the biomolecular components. That means protein, primarily protein and lipids. Because protein is the one that does all the functions of the body. Okay, and lipid is the one that's like oil, lubricating and making different kinds of things work together. So we do develop this Raman technique for liquid biopsy, and that's what we'll call Ramanomics. It's similar to like proteomics and genomics using Raman technique. And what we showed that we can take the overall spectrum and we can break them into the components of protein and lipid very easily by looking at comparing with the model spectrum sample, standard sample. And so we can get contributions of lipid and protein and do concentration profiling based on their intensities. Okay, and basically what we found, and the same thing we can do for RNA and others, and different organelles, nuclei, endoplasmic reticulum, mitochondria, in a cancer cell, in a normal cell. And what we found was that if you look at Raman concentration profile of lipid versus proteins let's look at the bottom line. So what you find, that you find very high concentration of lipid in cancer. Whether it's cancer cell or we took tissues. So cancer cell was in a suspension, so it's a body fluid. And tissue was a slice of tissue of a disease or a cancer patient. And if I, what happened now? Maybe it heated or something, or वहाँ का projection का problem. Is the lamp on? Or maybe the lamp needs to be changed, no? Okay, good, thank you. So again, what we found that if you plot the distribution of lipid versus proteins, the cancer samples, there are much higher level of lipids. So what we are proposing that we can use this elevated concentration of lipid as a biomarker, as a signature of cancer. So it's possible to add this to certain kind of antigen excretion, certain kind of proteins that are also expressed, add to that list to profile cancer. Okay? And so the next step is that we want to partner with a big company called Thermo Fisher to actually sell a product that is combines their hardware with our software and bioanalysis tool, for medical application. So this product actually already, if you remember, I talked about this special program from the government. It's called a Small Business Innovative Research, SBIR. They provide money to small companies to develop a product that is useful and commercializable. So we got one of these grants through a company that I formed called Advanced Cytometry Instrumentation System, and work with Thermo Fisher to develop this product for commercialization. Another thing is that we are also using not only intrinsic Raman, which is lipid, protein, and DNA and RNA, but I can also put, like fluorescent probe, I can put organic molecules that had a specific vibrational band which I can use for mapping out the distribution. So instead of fluorescence, you're using the Raman signature of that probe that has gotten in there. Okay? And you can make different kinds of probes like these, and you can look at vibrational band associated with this as a group, okay, as a probe, and do Raman imaging. But again, Raman technique is generally not as sensitive as fluorescence. That's why there are special things needed to enhance that. One method is resonance Raman. Resonance Raman is when your wavelength that you are using to use a scattering, that wavelength is very close to absorption band, electronic absorption band. then it's resonance enhanced. The scattering is much more efficient. It's called resonance Raman. Okay. The only problem is that most of these small molecules, their electronic transition is in a much shorter wavelength, like 400, 500. So or even in UV. You don't want to use shorter wavelength light for the, as I said, because of penetration or damage. So we are designing organic structure with bigger conjugation and all those, so that excitation will move to the longer wavelength, and we can do resonance Raman with the added probe. So that's another area that I would like you to think about, that you don't need to just make fluorescent probes. You can make a resonance Raman probes. You can synthesize organic molecules with a functional group that gives you Raman spectrum in the region where the biological material don't scatter. Like the bands from lipid, protein, DNA, or RNA are not overlapping, a clear region. Okay, and one of the group we have found is over here, this group. So anyway, these are some examples that the imaging of tissues, not cells. So here we have a mouse that in the pouch we grow tumor, and by 2-photon imaging we show that we can actually map out the distribution of cancer in the tumor site. Because what happens is that those dyes, when they get into the tumor, and they light up with the fluorescence, 2-photon fluorescence, and highlight the regions of the tumor. Okay? This is imaging of a tooth. Tooth inside has these channels. Okay, they're called dentinal tubules, long channels, tubules. And we showed that if we take a 2-photon dye and mix it with adhesive material that fill in, when you go and you have cavity and they fill in, or you have root canal, they fill in, then this bonding material, that filling material, the best one has to penetrate in dentinal tubule and bind, give you strength, okay? So we mix 2-photon dye with that, and the deeper they penetrate in dentinal tubule, the better they are. And we showed that actually we can image this tooth from outside. We send 800 nanometer light, and the emitter is in green that can come out, and I can image all these things, hundreds of microns of those tubes we can image. And tooth is a hard tissue. It's very scattering, very dense hard tissue, but still by 2-photon microscopy we could image it. This is application of OCT, optical coherence tomography. We work with a group in Brazil. We are not dentists, but we work with dentists over there, and we could show that actually you can use optical coherence tomography to look at caries and other little decays in tooth. Later on we will say that to enhance contrast in OCT, you need nanoparticles that scatter much more strongly, and gold nanoparticles scatter very strongly. So you embed gold nanoparticles in tooth someplace, we can have enhanced scattering and enhanced detection. So we show that too. Then in imaging, live animal imaging we also do. And live animal imaging, we can have nano formulation of emitters, whether it's quantum dot, we'll look at it in a few minutes when we talk about nano emitters, or organic dyes embedded in a sol-gel glass matrix, or enclosed in a polymer matrix. So these are all the nano emitters. So what we can have is, we'll see later, nano formulation, that these nanoparticles will have emitter. Then on the surface, through surface chemistry, we conjugate it to a targeting group, which is generally antibodies, some cases, And then they can go and look around for a disease cell that on the surface has these antigens expressed, overexpressed. So they said this cell is different from the normal cell, and they have affinity for that, these antibodies to the gravity. Okay, and then cell by a process called receptor-mediated endocytosis. It takes the nanoparticle inside the cell, and then you can image it, and you see how is the distribution of disease cells. So normally we take the mouse, we have grown the tumor that we want to target for imaging and therapy, treating, see how this tumor disappears. So over here it takes some time to grow this tumor. After that, we inject a nanoparticle into his bloodstream through tail vein injection. And then these nanoparticles can move around. Firstly, they go into the liver. Then they have to escape the liver. We'll talk about it later, how we make it escape, so on. And then eventually they come to the tumor. They recognize the right cancer, and they go and grab on there, large on there. Then we can image it and see what is the distribution of image, okay? So, firstly we inject it, then we partially anesthetize them. It gives anesthetics a little bit so that the mouse isn't moving around, right? And then you clamp it on a stage, and then we have this imaging system. So there is a stage there, we put the mouse there, and then we shine the light, and the light comes in there, whole body, and excites this fluorescence over here, and you can image. So in this process, you can actually trace how the nanoparticle is moving around. And that's what is called biodistribution, changing as a function of time. And this gives you what is people like to call pharmacokinetics. How the distribution changes in time is a very important pharmaceutical parameter for any drug treatment. How is the drug distributed and moves to the target? Okay, so we get the whole information in course of time. So here one is with quantum dots, which we have abandoned because cadmium is toxic. So we don't use anymore. We'll talk about other kind of quantum dots. And we injected it, and you can see the tumor lighting up after a certain time. When it localizes, it lights up over here. So we can do this imaging. Now most of you who are here will not be involved in this. I don't know how many of you have fear of working with mice, live mouse. Yeah, you have, yeah. And then you're trying to inject it in the tail gun. Poor mouse is jumping around, you know. And sometimes you have to wear thick gloves, because the mouse might bite you. Okay, so you need to wear thick glove so that it doesn't penetrate to your skin. Anyway, but I have gotten students who are chemistry students, and they are in synthesis, and then they slowly venture to do this. Oh, it may be fun, I want to learn how to do animal studies. And it's okay. As long as they're open and their mind is to do something like this, they can do this. So we encourage people to not limit your ability. You know, you could be multitasking, right? From synthesis to in vivo studies. Doesn't that sound great? Okay. So here is the in vivo tumor uptake. Again, there is a tumor. We can wait and see the emission. This is false color, ratio of tumor to skin. So you want to see also, skin also fluoresces. But the trick is that the dye you have put in there, or nanoparticle, fluoresces at a longer wavelength, so that you can overcome the emission of the skin or the hair. Okay? And these mice, sometimes if you want to be very clear, you can shave the mouse, the hair, removed. But most of the time, if you choose a longer wavelength, you don't need to shave. You can do the whole mouse by itself. You can do that, you know. So you can use either what is called nude mouse, as a hair shaved, okay. Otherwise, you can use a whole intact mouse also. So we do routinely this in vivo studies too. Now we will talk about it later, that people are thinking that it's not enough to do a study on mouse, because mice are not good example of a human. We are much bigger animal, right? Our heart and lung and brain, they're all different some ways around that. So how can we, what we study in mouse, how can we translate it to human? Okay, so there should be, so what is the closest relative to us? Huh? Rabbit, yes, but not exactly. Monkeys, yeah. So, there is a lot of push to studies in monkeys. Then there are some concern. Monkeys are our cousins and we should really experiment and sacrifice them, you know, and look at their organs and so on, right? And the second part is economics. It's very expensive to do tests on monkeys in America. So we did only one study on monkeys, and that was toxicity studies. We wanted to say if the nano formulation is less toxic or not. So we did it on monkeys, and that we did in collaboration with a group in China. Let Chinese kill the monkey, not us. And also it was cheaper to do it over there. Actually, in old culture, they ate the brain of monkeys. Did you know that? And it was very really repulsive way. The monkey is alive, and they put it in a chair and clamp it and open the brain and then eat the brain. Live monkey. So anyway, let's not get into that direction. So monkey is the best one, but as you said, yes.
So, but in a country like India, like targeting on using monkeys for experimentation, this can help.
But we worship monkeys. How are we going to kill monkeys over here?
It won't be that easy. It's
Yeah.
not the relative sentiments of the people.
Yeah. So I think again there are issues in each country. Here it is because we consider monkeys like God, and you don't want to kill them, and so then you may think that. Huh?
You can easily kill man here, but not
Monkeys, right? Same thing. You know, in our area, you wouldn't believe. In our area, when I was growing up, we also had snakes. We still have, but they don't wander around the houses because the population has grown so much. But I remember in my house, a couple of times cobras coming in there, and nobody wants to kill the cobra. They want to put milk and worship and ask them to get out of the house. You know, they got some people to kill finally these. So anyway, there's also a problem of killing snakes, right? Huh? Yeah. So what happens is then for certain studies you could use pigs. But again, I don't know, either Hindus or Muslims would like to kill pigs and experiment. That's another issue. So go from mice to, as you say, rabbits, or firstly, rats are bigger. And then you can go to frogs or pigs trials are very complicated. First, you have to get permission from the Food and Drug Administration. And it's okay. You show all the data, everything, and not one is on the statistical significance. That means how many times you can repeat the experiment the same. You do only with 2 samples is not enough. Okay. So you need to show that, and then you get permission for human trials. And human trials are very expensive, from hundreds of millions of dollars. Cannot do it. So once you get a drug or nano formulation something, and you have advanced up to the animal level, everything is looking very clear, then your hope is that some pharmaceutical company will become interested, license your technology and run human trial, because they can afford it. Academic people cannot afford it, or even a small company cannot afford it. Okay? So anyway, these are studies from animal. Then we are looking at sensors. Sensor is detecting certain components. And one example you already saw, looking at profiling using Raman spectroscopy, but in sensor, you are looking at the response to physiological changes or response to some kind of ion activity changes. So in general, global changes that may occur as a function of disease or something else, whatever. So you want to detect that and how the progression stage. So that's what is biosensors. So biosensors is detecting. And when you combine sensing and imaging, then we call that thing functional imaging. It monitors not only spatial distribution, but also monitoring how the functionalities like metabolic activities and others are changing together in different domains. So that's called functional imaging. Okay, so let's talk a little bit about how long are we going to go, Dr. Ghosh? 1 o'clock? How is what? Huh? Half an hour more? It's okay, or are you tired? No?